phosphorescence in water.
Wonderful. So now on to tonight's program. As many of you may know, I am big, but I am not Big H, also known as Harold Lavender, which I'm sure many of you know. Harold unfortunately could not join us tonight. He is in Los Angeles visiting his children, but he wanted to pass on a message tonight. He said Dave Hovda and he worked together at the Arroyo del Oso golf course in the early 1970s. Apparently, Dr. Hovda caddied for him in several tournaments, and the skill in his difficult job clearly helped define his future academic successes. The caddy fee of $1 was obviously was put into your college fund, so he appreciates you for all the help there, and he welcomes you back to the University of New Mexico and wishes the best for you. So,
thank you very much.
Sure. Just to go into a couple points here, so Dr. David Hovda is a proud three-time alumnus of the University of New Mexico. He is the director of the UCLA Brain Injury Research Center and is a past president of the National Neurotrauma and International Neurotrauma Societies. In 2016, Dr. Hovda received a Lifetime Achievement Award from the National Neurotrauma Society. Additionally, Dr. David Hovda Research Award was established to recognize the highest rated high school, undergraduate, post-baccalaureate poster presentation during the annual National Neurotrauma Symposium. He has consulted with Major League Baseball, the National Football League, National Hockey League, and the World Boxing Council. He has supported the Veterans Administration and the Department of Defense. In 2012, Dr. Hovda received the Alumni Association's James F. Zimmerman Award. One of our highest awards is the Alumni Association. So if you would, please put your hands together for Dr. David Hovda.
Thank you. Thank you very much. So Big H couldn't come tonight, huh? So is everybody here a Lobo? Woof, woof, woof. Either that or you get an 8 count from UCLA, you know. So I did caddy for Harold Lavender a couple of times. And I remember this par 3, used to be called the South Course. Now it's called the Championship Golf Course. And he turns to me, it's a long par 3, and I know he's going to watch this later. And he turns to me and he says, Dave, he says, what club should I use to hit this par 3? And I said, Laddie, I've seen your golf swing all day. You haven't got a club in the bag that'll make it. A little bit later in the presentation, you'll see Harold Lavender's golf swing, and I'll point it out to you. Since this is about brain and brain injury, that kind of goes together, you know. So seriously, one of the things that I have got to say is how grateful I am to Kim Feldman for inviting me to come to speak and to come home, to come back to my roots. You know, the University of New Mexico has done so much for my career, and I got such a wonderful education. I wish more people had the opportunity I had by coming to this wonderful institution. At UCLA, when I left here, I went to UCLA in 1985. Actually, 1984 and 1985, it was kind of a crossover with the idea of just staying for a year or so and then moving on, because I didn't think I was going to be smart enough to stay there. I'm still at UCLA, and I'm going to be retiring from UCLA. And so I've done something that I guess is okay as far as UCLA is concerned. The Brain Injury Research Center was started in 1990, and it was recognized as a state of excellence, center of excellence by the National Institutes of Health for traumatic brain injury and recovery of function. So I'm going to divide this lecture up into some very similar, very simple parts. First part's going to be what a traumatic brain injury is. You all know how to beat somebody on the head, all right? My job is to tell you what happens inside the head when you beat somebody. And then the second is I'm going to tell you a little bit about the basic science of what happens to the cells inside the brain. Very small, very short. No formulas, I promise. No exams, nothing like that. There may be one diagram that's a little crazy, but it's important for you to see. And then we're going to talk a little bit about what concussion is and why concussion is should be considered much like a traumatic brain injury. And at the end, I can talk to you about the long-term effects of mild traumatic brain injury or repeat concussive blows. And we can talk about the definition of subconcussive injury and whether that really exists or not. And then I'm happy to answer any questions that you have about the National Football League, about playing soccer, about heading the ball being in soccer, about athletics, about whether, if you had a son, would you have your son playing football? To get it off the table, I played football. I was never good enough to play for UNM, but I did play, football. I was a middle linebacker. I was never taught to put somebody gently down on the ground on a draw play. I was told to kill somebody as they came across the line of scrimmage. So with that said, you're going to see both animal work, some basic science work, and some clinical work. And then we're going to combine it all together at the end, and we're going to make some general statements about mild traumatic brain injury and what it's about and how we should approach it, both from a sports standpoint as well as from a healthcare standpoint. So first, disclosures. I'm a tenured professor. I don't have any stock. I don't care about the National Football League. I've never been charged with sexual harassment. I don't do anything fun, I don't do anything weird. I'm just a professor, okay? I don't care if they kill each other on the football field or on the soccer field or anything like that. One thing that for the students, for those of you that are students or young professors in the audience, I think it's really, really, really important that you acknowledge the shoulders that you stand on. And this person up here at the very top is still alive. His name is Dennis Feeney. He was a professor here in psychology. He introduced me and a guy by the name of Gordon Hodge to physiological psychology. And from that moment on, I fell in love with the brain. And I didn't know how I could ever study anything else except the brain. This person here was the chairman at Johns Hopkins for neurosurgery. His name is Earl Walker. He retired and came to New Mexico and became a professor emeritus. And he and I did studies together on the definition of cerebral death. And I'm happy to talk to you about that, but that's a different lecture, all right? I went and did my postdoc with this man here. He's a neurologist. His name is Jaime Villablanca. And there I learned cerebral hemispherectomy. That's the removal of half of the brain for children that are just born, that have infantile epilepsy or Rasmussen's disease. And you can remove half of their brain, and you know what? They go to high school, they go to college, they become doctors, they become administrators, they become lawyers. But if you do the surgery too late, there's not enough plasticity left. And I got a whole set of lectures for that too, but we don't have time to talk about that. This person here, Donald Becker, he came to UCLA the same time I did, and his specialty of interest was traumatic brain injury. And I went to the emergency room and I saw my first traumatic brain injured patient there, and I realized that they needed something completely different than what we were able to do in the laboratory as a basic scientist. And it was frustrating to me in the 1990s that we had nothing to offer anybody that had traumatic brain injury. There's no treatment for recovery of function. Rehabilitation, physical therapy, and occupational therapy help. They improve the quality of life. But there's no drug that I can give you. There's no experimental neurogenesis of cells that I can give you to repair the brain. So what is traumatic brain injury? Well, it's really caused by a mechanical force, biomechanical force. I can't tell you the number of times people have come to me and they say, well, I've had a head injury because I've had a stroke or I've had a seizure or something like that. Traumatic brain injury is like a baseball bat. You get hit by a baseball bat, or it's an assault, or it's biomechanical in its nature. It's not stroke, it's not ischemia. It can produce cell death, but in many cases it produces a regional and temporal cellular alteration of function. It produces a state of energy crisis. Energy crisis? How much energy does it take to run the brain? I'll teach you, I'll tell you. It's really cool. And a subsequent thing called metabolic diaskesis. Well, what the heck is diaskesis? Well, that's a fun term that was used by a long time ago. It's misused today by neurologists, but it usually means, it literally means shock throughout. And it means that something that's going to recover eventually. You show deficits and it will recover. And I'll teach you what that means. It changes the priorities of fuel. The brain, all of a sudden, doesn't use the same fuels that you thought it used normally after brain injury. And it causes the acquisition of post-traumatic stress disorder or neurological degeneration that's related to different diseases. All kinds of diseases, not just chronic traumatic encephalopathy, which you've all read about. This is a guy named Lee Goldstein. He's a friend of mine from the University of Boston. He sent me this from New York. And this is a papyrus from an Egyptian physician. And I'm sorry, I can't interpret it for you. I have to take their word for it, you know. But it just simply says it's really important to understand if somebody gets hit in the head because they're different after they get hit in the head. That's what it means, okay? So we've known that this is a problem for a couple of thousand years, okay? Konstantin von Monaco worked with a guy by the name of Broca, or not with him, but at the same time. And Broca was the man who discovered that language was located in the left hemisphere. Well, von Monaco would have people that had strokes in the right hemisphere, and they couldn't talk for a while. And then all of a sudden they could talk again. And so he wanted to coin a term that was temporary, that caused a temporary dysfunction that you slowly recovered from. And he called that term diaskesis. And he did this in the early 1900s. Earl Walker, in 1944, wrote what I think is the seminal part, a seminal paper about concussion as a mild traumatic brain injury. And when I gave the talk to the National Football League in Hamilton, Canada, I learned a phrase that I thought was very cool. And that was, I don't know what is so mild about mild traumatic brain injury. You know, I just don't. So let's talk about energy. I told you energy is important, all right? So you run at about 522 amps per second, all right? You make about 65 kilograms of ATP a day. That's energy. That's a type of energy, adenosine triphosphate. You consume about 380 liters of oxygen per day. The average human burns at about 116 watts of power per hour. That's kind of like a dim light bulb. Now there's this junior college in Las Cruces. Having gone to Washington, D.C. several times, I can tell you there's some pretty dim light bulbs I've seen. You know. So what happens when the brain moves? Well, you know, the brain, people take a model of a brain, of a human brain, and they'll say, yeah, this is a human brain. It's about 360 grams. You can play football with it, and tuck it out in your arm, that kind of stuff. And it's always rubber, you know. And even in the gross anatomy lab, when I would take the brains out of the skull, they'd be fixed and they'd be hard, and they would have them in a jar. And the students would pick them up and hold them, and they'd play with them. But you know, if I take you to the autopsy room, and I take a brain out of a human just from the autopsy without fixing it, it's like undone jello. It'll almost go flat. It's like undone jello. And when it's moved violently, it's not moved in one like this and back. It's moved the same way that this finite element looks like, where you have different parts moving at different degrees. And that's because different parts of the brain have different consistency. When you start putting your finger through the human brain, you'll have different parts of resistance. And that's simply because some of it's thicker than others, okay? And then consequently, it will move differentially than others. And when it moves differentially, that causes the stresses and strains that pull the brain apart and pull the cells apart. So how do you do this in an animal model? Well, it seems kind of ridiculous to take an animal and take a hammer and bang it over the head. That sounds real scientific, doesn't it? Doesn't sound very humane. So many years ago in Sweden, they just discovered a certain way to do this. This was actually discovered at Wayne State University by a bioengineer. What you could do is you could take a small squirt gun. So this is a cylinder that's filled with saline, and this is a hammer that you can drop down, hits a piston, generates a fluid pulse, and then it generates, and you do a little hole in the rat skull, and it pushes a little bit of saline into the skull, and it moves the skull, moves the brain at a certain distance, at a certain velocity. So those of you that remember physics, it's called work, which is force over distance. But now we're going to divide it by time. All right? Now, when I was here, I asked a lot of girls out for dates. I did, man. I worked it hard. I was a member of Lambda Chi Alpha, and I worked up and down the street, and I asked them out. And every single girl would come to me, and they would say, no. No, man, no. I didn't give them a concussion. But if I had Mike Tyson move their head that same distance, over 21 milliseconds, they'd be out like a light. So it's not necessarily the movement, it's the degree of work that the brain actually receives. Well, if you drop that hammer at different degrees, what happens? Well, if you make the concussion very, very mild, and then you increase it, and you measure what happens inside the brain, what happens inside the brain is the brain releases a chemical or an ion called potassium. And Leao discovered this in the 1940s. And it causes a depolarization. It's very, very brief. It only happens, if you look at this slide here, it shows the increase in potassium that goes up. It only lasts for a couple of minutes after the traumatic brain injury. But you'll notice that compared to sham, you have to get to a certain part of severity, and then all of a sudden it happens. And then you get this release of potassium. And we thought, oh my goodness, if you have this release of potassium, Mother Nature doesn't like that. And she would like to have that potassium go back into the cells so that the cells can fire again. Because if the potassium stays out of the cells, outside the cells can't fire. And they become depolarized. And that's called spreading depression. So we thought, well, maybe the brain requires energy to activate these pumps. So Louis Sokoloff in 1977 discovered how to do this. And this is a slide of a rat brain. Here's the arrow showing the fluid pulse. And the red is the increase in glucose use. And the glucose is burnt. And so we have called it, the burning of glucose to concussion or traumatic brain injury. And it burns at 200 times the amount that the normal brain would work. It's an enormous amount of glucose that's burned. And glucose is a fancy term for sugar, is the primary fuel for the human brain. Now, we thought, well, gee, I wonder why we did this, what happens? Well, when we do this in the animal literature, if you look at this hippocampus here, you can see that it's burning really hot. It's really hot with glucose utilization. But if I cut the fibers, which are the glutamatergic fibers, the brain, the hippocampus isn't black. It actually is quiet. And actually, if you measure what happens, you actually see an increase in lactate. Well, if you remember, if glucose is burnt and you don't use oxygen, or you don't have enough oxygen, you make something called lactate. And you make something called lactic acid. Have you ever tried running up the stairs, like at the pit? When we used to have seats. We were kids. In the student section, you used to have seats, And now they don't, which I find odd. But anyway, so if you run up those stairs, the first thing that starts to happen is your thighs begin to burn. And you wonder why that happens. Well, it's called lactic acid. And so lactate will be formed in the brain. Lactate also is a signal in the brain. It's an important signal, and I'll tell you what happens. So in cartoon fashion, let's see what we do here. So let's take our hammer. We hit this neuron, okay? All the cells in the brain fire. It's like having a mini seizure. And the cells release glutamate and fire, and the glutamate binds onto a receptor, and potassium is released outside the cell. And Mother Nature goes, this isn't good. We got to get the potassium back in. We're not killing the cells. We're just calling them, we're just making them dysfunctional. So what happens is we have this pump called the sodium potassium pump. And Mother Nature says, okay, well, we'll take this glucose, we'll make some ATP, we'll drive this pump, and we'll pump the potassium back in, and everything will work just fine. And this is the first part of the energy crisis that occurs following a concussion, is that the brain needs to burn. It needs this glucose. So we began to say, well, what if happens over time? What if we do this and we study these animals over time? So when we do that, we find that, in fact, if we do it at time 0, we give the animal a concussion, you see this red area burning. Then at about 1 day, the brain becomes depressed, and it's like it's exhausted. It's burnt so much it's exhausted. So it stops, and it rests. Mother Nature has a chemical that's released. It's called adenosine. It says, I want it to rest. And then it will slowly start to increase, and it will recover back to normal. And while it's dysfunctional, during this time, the animals will show cognitive and spatial deficits as well as motor deficits. But you know something? It's not how hard you hit the brain. This happens whether you hit it severely or you hit it mildly. It's the time that you spend in these states that's related to how severe the injury is in the animal. Well, potassium is not the only ion that we're worried about. In stroke, we've known for years that calcium, if that comes into the cell, you can kill the cell. Calcium is hundreds of times higher in terms of concentration outside the cell than inside the cell. So if you open it up a little bit, the membrane, it comes rushing in, and calcium will come rushing in. And what happens is calcium will rush in for 4 days. So you have potassium, which releases for about 2 to 3 minutes, and calcium, which goes in for about 4 days. This happens, there's no cell death yet. We're still not talking about cell death. We're just talking about a dysfunctional neuron or a dysfunctional cell. And we're talking about a vulnerable cell. So now we have this cartoon again where we have calcium coming in, and the neuron, Mother Nature says, you know something, I don't like this calcium. This calcium is going to kill me. So you know what she does? She's really smart. She says, I'm going to take the mitochondria and it's going to capture the calcium. I'm going to buffer the calcium. So it's going to go to the mitochondria, and all of a sudden you can't make the ATP. You can't make the glucose. So now you need the energy. Calcium comes in, shuts the ability of the energy to be made. And that is your, if it gets bad, it can cause proteasis and cell death, and that actually causes more dysfunction. In the axons, this can happen as well. Calcium can come into the axons, and you can have axonal swelling and inflammation, and this is the way cells communicate to one another through the axons. And this can actually cause microtubule damage and neurofilament damage and plebs. So what happens is you have a neuron now that is vulnerable because it's very weak, because it's trying to recover from this blow that you gave it, and it also can't talk to its neighbor. That's why people have deficits. That's why you have amnesia. That's why you have unconsciousness. That's why you have things that are a problem following a mild traumatic brain injury. Well, that's not just the only bad thing that happens. If you take a normal animal and you stimulate the cortex, you can have an increase in glucose metabolism, and you have a corresponding increase in cerebral blood flow. The brain doesn't store glucose. It doesn't store it. It gets it from the blood. That's why magnetic resonance imaging works so well for functional magnetic resonance imaging. And so what happens is when a cell fires, it asks for more blood, and then the blood is coupled to the energy demands of the tissue. In this particular case, in the normal case, there's no cell death. This is the way we all work. We work on this every day. This is when we read, we look at lights, we listen to a lecture, we go to sleep when somebody's lecturing, you know, whatever happens. But after a concussion, up to 3 days after a concussion, I can stimulate the brain, I can still get an increase in glucose, but I don't get an increase in blood flow, and now the cells die. So they die. So I have a vulnerable brain. If I stimulate it while it's vulnerable, it can't make the energy that it needs. It can't survive the signaling because the signaling pathways are gone. And now you're starving it to death because the blood flow doesn't get to the part that it needs. If you plot all this out, this is an enormous amount of science, and it's not just us, it's across the world. This happens to be a neurochemical metabolic cascade, and this is in a rodent, in a rat. And this is in minutes, hours, and days. And these things, calcium, potassium, lactate, blood flow, glucose, all these things happen at different time courses. So it's not that it happens all at one time at the same time. That's why one chemical or one bullet, there's no silver bullet for traumatic brain injury. It's going to take a multiple compound, or it's going to take multiple treatments. And this is what we've termed the cerebral metabolic crisis. When calcium comes in, calcium will bring in sodium into the cell. So those of you that had breakfast or lunch today, and I'm married to a cardiologist, so I'm doomed. But you go and you put a little too much salt on the French fries, and you eat the French fries, and your fingers begin to swell. Same things happen to the cell. When the cell gets sodium inside of it, it swells, and that's called edema. That makes a tight brain. So now inside the skull is a closed box. This is a closed box. This is your helmet. It's closed. If I open it, and you come to my house, I serve you peanuts on this side. On this part, it's got ridges in it, okay? But the point is that it's got a closed box except for this hole. This is where the brainstem comes out. If the brain swells and it's closed, what happens, it'll swell, it'll try to come out the foramen magnum, and it will crush, it's called herniation. It can crush the brainstem and it can cause death. And so this hole here, which I just showed you, is the exit for edema. So you lose a few neurons. What's the big deal? When I started medical school here, graduate school, I was told, you know, you're born with so many cells. You have a martini, you knock off a couple, you know. You got 1 million of them. You know, what's a few cells that are gone? Well, let me show you what happens. I'm going to show you Harold Lavender's golf swing. You're going to see a line of guys in a driving range and gals on a driving range, and they're hitting golf balls. Think of it as a neuronal network, a network of cells that has a function. Okay, and then we're going to bring in a neuron, another person, and I'm going to show you what happens. See if this will work. Yeah, so here's the network. So these people are hitting golf balls. Harold's the third one from the left. And along comes a real important neuron. And the neuron next to him sees this guy. And he goes, God, did you see that? That must be wonderful. So he tees it up. And he says, you know something, I'm going to hit the same thing he does. So he hits it. Harold's frustrated. Harold sees this. So Harold says, well, heck, I'm going to swing same way. So he swings same way. So this goes on and on and on, all the way down the line until they're all swinging. And they're all making beautiful music. And then all of a sudden we have a concussion, and this neuron becomes dysfunctional and leaves. You know what happens? cute, isn't it? When is it time to return to play, and when does the repeat? What happens in repeat concussion? Well, concussion, football is getting a lot of press, but concussions happen in boxing, they happen in cheerleading, they happen in lacrosse, they happen in field hockey, all kinds of different sports. There are 44 million annually children and adolescents in sports in the United States. 6.2 million of them are high school athletes. 340,000 are college athletes who are going to get concussions. And 144,000 are pediatric concussions that go to the emergency room. Okay? There are 2 million Americans that are going to get a head injury this year. 2 million. So what we did is Amy Prinz, who was my graduate student, began to look at this metabolic cascade. Now here's this glucose spike, and it slowly begins to recover. It comes another one that came along. Well, when we give an injury during the time that it was cells beginning to recover, it now takes longer to recover from an injury, and cells die. So we became very worried that repeat traumatic brain injury can cause other types of problems besides just long-term problems in injury. So we saw now repeat injuries, we saw cell death along the ventricles in the animals. We saw a change in the decrease in body weight and delayed puberty in very young animals. We saw a decrease in growth hormone. Today, when you have a head injury and are recovering from the head injury, you now are tested for hormones, primarily growth hormone as well as testosterone. Because testosterone is also decreased for a long period of time. And the sexual behavior, your libido changes after traumatic brain injury. For a 16, 17-year-old, that's a big deal. Now, I have not met any rat that's a human. I have met several humans. Are you listening, Harold? Which are rats. This is Mr. Gonzalez and Mr. Vespa, Dr. Vespa, runs our intensive care unit. This is our PET scanner, and this is how a PET scan works. What you do is you take an atom and you add a proton to it. It wants to get rid of that proton. It runs into an electron. When it hits the electron that's neighboring it, close to it, it gives away 511 K gamma ray, and it's detected so we can tell where it happens. So we can measure glucose or oxygen or blood flow in human patients without killing them, without doing the same thing you do in the animals. So for this experiment, I'm going to show you, this is just a color bar, just for your reference section. So the lime green is about 7.5, all right? This is a girl that came in, I'll never forget her name. Name is Chapman. I have her permission, her family's permission to show these slides. She was an 18-year-old female in a motor vehicle accident. She was hypoxic at the scene. She was a Glasgow Coma scale of 4. She was very close to cell death, very close to death. And we did a PET scan on her. Look at a burn. Look at the red area at the top on the top part of the slide. That's burning. She lived for 9 years in a vegetative state before she died. And when we did this over this time course of patients, we saw the same thing we did in the rats. We saw an increase in glucose metabolism, a depression, and a recovery of function over time. Again, we were concerned about whether it was a degree of severity. If I look at these images, these are glucose images. This is cerebral blood flow. This is oxygen. This is oxygen extraction fraction. This is all one patient. It's not the color, it's the intensity that's important. They should be matched. The same parts that are really bright red here should be really bright red or white here, should be really bright green or red or white here. And they're not. We've lost the stoichiometric relationship between glucose metabolism and oxidative metabolism in human beings the same way we do in animals. This oxygen extraction fraction is the amount of oxygen that's taken from a unit of blood as it goes through the brain. This is not cerebral ischemia. This is traumatic brain injury. And then this happened. This scared me to death. So I had the coach, the athletic trainer for our football team. Our football team practices on campus, doesn't practice at the Rose Bowl. And Jerry Feinerman, who was the physician, called me and says, "Dave, I got this kid that had a concussion. Would you like to image him?" And I said, "Yeah, I'd love to image him." So I happened to have at the same time a woman, a person that came into the emergency room with a severe traumatic brain injury, and I had a slot open the next day. So I went to the practice field and I got this guy, and I talked to him. I tried to remember everything that I learned in the Department of Psychology here at UNM. Who's the President of the United States? Who was your girlfriend? What classes were you taking? What do you want to do? And he said, "I don't care what you do, Doc. I'm happy to help you with your research, but don't show this to the combine, because I want to play professional football." So I went out and I found a normal UCLA undergraduate student. That's kind of an oxymoron. And I ran a PET study on him. There's his PET, that's what a normal PET study looks like. So I did the two people, one that had a concussion, the other one that had the severe traumatic brain injury, same age. And I called the technician that night and I said, "Show me the concussion patient." She said, "That's the concussion patient." I said, "Nah, that can't be the concussion patient. Show me the severe injured patient." She said, "That's the severe injured patient." It doesn't make any difference whether you're Glasgow Coma Scale of 15, which is normal, or 5, which is severely injured. This is a biomarker for mild traumatic brain injury. This is a way of looking at a picture of concussion where the two things, the one thing that's very obvious that happens to people that have concussion on the football field, any athletic trainer will tell you this. Any doctor will tell you this. Any coach will tell you this. Athletes lie. "Are you okay?" "I'm ready. I'm ready to go back in." Same with military personnel. In Europe, Vagnozzi did the same work, only he did it in people that played soccer and rugby. And he looked at patients with NMR spectroscopy, and he looked at NAA to choline ratio, which is a measure of mitochondrial functioning, and noticed that in the frontal lobes of a single concussion, it took them about 30 days to recover. So they did the right thing. They took these players out of play, told them they couldn't play for a while. A couple of them sneaked back out, played, got hit again. So then he did the same study, looked at it, now it takes 45 days for them to recover, just like in the animals. So when you look at a head injury that has a right contusion, and you look at a spectroscopy, it shows a lot of lactate, and you have a microdialysis probe here, you can see there's no glucose, but lots of lactate. The brain has burned every single mole of glucose it has, but it's not getting enough oxygen. It's making all this lactate. The same thing that's burning your thighs as going up and down the stairs. If I put it in, look at a different region of the brain of the same patient, I don't see any problems with lactate. Lactate's very low in the microdialysis probe. I have plenty of glucose. Head injury is regional. It is not one organ. It happens differently to different parts. You can have a concussion of a temporal lobe and not a concussion of the parietal lobe. Okay? They would have different time frames in terms of their recovery function. Well, maybe we ought to just give people a Snickers bar when they come off the football field. Maybe that's the reason we have such a lousy god dang. I'm always watching the paper looking for the scores, for the UNM football team. I wonder why that is. Maybe we ought to give them Snickers bars. Patients that come in with a head injury are typically hyperglycemic, meaning that there's a lot more glucose in their blood. And Mother Nature does that. And we just wanted to know what happened in the brain when we gave these people serum in order to reduce the amount of glucose in the blood. So here's a person that comes in with hyperglycemia that's around 300. We give them the start of insulin infusion, And then the glucose goes down to normal, around 100. But look what happens in the brain. It goes to 0. We're starving the brain. We're hanging the wrong thing on the IV bag. Same thing with glucose metabolism and positron emission tomography that measures blood flow. So say, for instance, we change the serum glucose around a big range in normal people, well, blood flow stays pretty constant. But look what happens in people with brain injury. All of a sudden, they show ischemia. So they're not even getting the blood we're trying to give them. Well, what about, maybe it's not using glucose the same way. We'll take 1 mole of glucose, take it down a vessel, have it drop down to a mitochondria, and for 1 mole of glucose, Mother Nature needs 5.6 moles of oxygen. Otherwise, you get lactate, you get that burning of lactate I told you about. That's called the oxygen-glucose ratio. In normal patients, or normal people, we'll have low glucose, lots of oxygen. In head injury patients, we have hyperglycolysis. And then we have something weird. We have very little glucose, but lots of oxygen being burned. For a matter of time, what does this look like when we do an image? We'll take a glucose metabolism study and an oxygen metabolism study, and we'll fuse them, and we'll get an oxygen-glucose ratio. Well, I can explain low oxygen-glucose ratio. That's when you're running up the stairs and you're making a lot of lactate. I can't explain high oxygen-glucose ratio. That means you're burning something else besides glucose. What on earth is there left to burn? So we decided to tag it. So we tagged glucose and acetate to see how it's being used in both neurons and in glial cells. And we found that in fact the glucose isn't being used the same way. And in human patients, what happens is that they're using the glucose to make things like proteins, to turn on different types of proteins associated with DNA, to help with the repair process. They're sparing them their energy for this type of recovery as opposed to trying to make it so they can work again. So you can't get the fuel there. If you get it there, it's not being used for the same thing. So people began to say, oh, this is really weird. You've got glucose that's being shunted out the wrong way, you've got lactate that's being used inappropriately, and you've got other shuttles that go on. This could be an opportunity, though, for metabolic therapy. Maybe for the IV bag, maybe we should be giving it some lactate. Maybe we should give it some pyruvate. Or when you were children, when we were born, what you did is you burned ketone bodies. Maybe we should give them beta-hydroxybutyrate and let the brain burn that. Then we started to look at long-term problems following concussion. And what happened during the Afghan war and the war in Iraq is that they had this silent epidemic, or this invisible wound, called post-traumatic depression or post-traumatic stress disorder. Well, head injury doesn't give you post-traumatic stress disorder. What happens when you have a head injury and you're a concussion, first thing noticed is you have amnesia. You can't remember. If you're a psychologist and you want to know what post-traumatic stress disorder is, you perseverate on what you've seen. You were there when the towers came down in New York. You were there when your buddy lost his limb. You were there when somebody got shot at a school system. That's what gives you post-traumatic stress disorder. So I designed a simple study. Got together with Michael Fanslow, and I said, listen, what we want to do, I want to do some Pavlovian conditioning for fear conditioning. So we're going to take an animal, and we're going to pair a sound and a light to a foot shock, so that the animal knows that if he hears the sound or this light, he's going to get a foot shock. And we're just going to look at the animal freezing. We're going to give him a concussion, and he shouldn't be able to learn this. Lo and behold, when we gave them the concussion, not only did they learn it, they learned it faster. Not only did they learn it faster, it was more unresponsive to therapy. Then in 1949, All of a sudden at King's College, this guy by the name of Critchney decided to coin this term called chronic traumatic encephalopathy. And he did this because of soldiers that were the effects of high explosives on the central nervous system. I don't know how many of you are fortunate or unfortunate enough to have fired an M16 or been next to a cannon, but it's like a pressure wave that comes back and moves you. And they were worried that that's what it was causing it. And it caused these long-term effects. Now we know from all these studies that came in the press from the University of Boston, dementia, Alzheimer's disease, violence, psychological problems. Well, even Lou Gehrig's disease, amyotrophic lateral sclerosis, this is Lou Gehrig, has been thought to be involved with this. So we decided, well, you know something, let's get really smart. Let's tag something that would be helpful in helping us identify Alzheimer's disease or something that would be akin to that called tau, tauopathy. So we tagged it and we used it with 5 retired football players. Gary Small did this study. These are the 5 retired football players, and you can see the red areas are where the tau is, and now in the control there's no tau. Well, there's some problems here with validity and reliability and amount and rate. And so just this year what happened is Zamalo decided to join us. We ran the same study on an individual who was 61 years old, played the NFL for played professional football for 30 years. And all of a sudden, we got his brain, and the same tauopathy matched exactly where the signal was coming from in our PET study. We may be able to scan people and tell them, you know, buddy, it's time for you to think about retiring, because you're going to have this. Now there's a recent paper that came out real recently that says that, you know, people over 60 and 65 begin to show this tau normally. This is part of the aging process, so it's still debatable. Now on the left here, this is an automated modeling of 56 different regions of interest of the human brain. So we do this at UCLA where we take these MRI images and we take them down to Art Toga, who's now at USC, and we measure the volume of the brain. Now, when you're born, the brain is small. When you start to grow, the brain gets bigger. And the French were right. Women's brains get bigger, mature faster than men's brains do. And then once you get to be 40, the brain starts to get smaller. It starts to atrophy. That's normal aging process. So I took this particular image of an individual, and I took him to a neuroradiologist, and I said, I overlaid the PET on it, and I said, can you just read to me and tell me what you think this is? He says, this is like an 82 or an 85-year-old person's brain. I said, this is a 22-year-old staff sergeant that just came back from Iraq. What about boxing? Boxing does this. Well, we look at this tensor imaging. You can see normals. You can see all these fibers that we talked about, these axons. They look pretty healthy. Here's a fighter that's been fighting for years, and look at the axons that start to drop off and that die because of this. And for some reason, women have decided they wanted to become fighting. And all of a sudden, we've got this thing called this cage fighting. Have you ever seen that on TV? I tell you why. I've got some people at USC who I think should go into one of those things. And then we had one of these, and then what we decided is how do you divide this up? If you take a bicycle wheel and you divide this into a left and a right hemisphere, and you take all the different regions of the brain, and you see the connections, and this is a normal individual. And after a traumatic brain injury, all of a sudden, these areas start to get bigger or smaller, and the connections start to fall away. And we published this a couple of years ago, and we're starting to use this. It's the same map that's used for genetics. So what is TBI? It's an injury that's caused by a biomechanical force. It's not stroke. It's not ischemia. It causes regional and temporal cellular alterations. It can produce cell death. It produces a state of energy crisis and subsequent metabolic diaskesis. We all now know what diaskesis is. It changes the priorities for fuel. Listen, people, we need to change our thinking here. This DEX-5 is not cutting it. It may be cutting it for the liver and the heart, because these are multi-trauma patients, but it's not doing anything for the brain. It can contribute to the acquisition of post-traumatic stress and to chronic neurological degeneration related to different diseases. Maybe traumatic brain injury shouldn't be thought of as an event that you recover from. Maybe it should be thought of as a disease that you're going to have to work with for a while. In JAMA, on February 1st, 2018, just a few days ago, when a sports-related concussion is suspected, the athlete should immediately be removed from play and assessed by a physician or a licensed healthcare professional. When in doubt, sit this person out. When I worked with the military or the athletes, I say the best person that can tell if somebody else has had a concussion is not the person that's had the concussion, it's the person next to him that says something's different. Tell coach, tell the athletic trainer. Get that person away, at least until he's stable enough to begin to exercise. I'm just a cheerleader here. There's an immense team that has helped me over the years, and I am so grateful, and they've done such a great job. And I hope if you ever get a chance to come to Los Angeles, I'm not in love with Los Angeles, I miss Albuquerque. But UCLA is a beautiful campus, and you're welcome to come by and visit. Thank you very much for your attention, and I'll answer any questions you have. I've got a bad back, so if you don't mind, I'm going to sit down. Not bad for 65, huh? Okay.
Doctor, is there a test for CTE right now?
Yeah, there sure is. As soon as you die, I can get your head, I can get your brain out. I got a stain that'll make you want to walk from here to Kansas and back.
Is there some way to be able to diagnose it now without having to die?
No. I have to say no. The FDA has not approved it. The ligand that we have, Pittsburgh has one as well that's working on it, to work with PET, positron emission tomography. And not every hospital or university has a PET scanner or a cyclotron to make it. What's important is that this tauopathy, or this tau, is a protein that's like a scaffolding that you would put on the outside of a building you're going to work on, but it's on the inside of the cell. And so we all have tau. What we're worried about is when it becomes phosphorylated, it bends on itself. And then all of a sudden, the structure of the cell starts to get weird, and as we start to have plaque and tangles, and that's called Alzheimer's disease, all right? If you just have tauopathy, we now call that chronic traumatic encephalopathy. So now we've got these two ligands that seems to be able us to see tauopathy. The problem is I don't know how much you need to have to be clinically relevant. And the second is I don't know why it moves. Apparently, in people that we've studied, it seems to move. It moves from the deeper part of the brain to other parts of the brain. And I don't know why yet. We'd like to know why. And I would think in about 5 or 10 years, you're going to be able to do, at least do more for what I call the stool of concussion. The stool of concussion has got 3 legs. First leg, do you have a concussion? The FDA just approved a serum test, so I can actually do it on the sidelines and see if you've had a concussion, according to the FDA.
How long does that take to get the result?
According to what they have told me on the press, minutes. I don't trust the FDA. There could be some dim light bulbs up there. I'd like to see a little bit more of that data. I saw some of it a year ago in Florida, and I was not impressed because the signal to noise, it's great if you have a severe head injury. I mean, if I put a hole in the brain, I see a lot of this S100B and this stuff in the serum. I don't need that kind of a test for the hole. When Gabby Giffords got shot, there's plenty of that in her bloodstream. What I needed was somebody with a close concussion, and that we don't know yet. The second leg of the stool is when can I say to this athlete or this soldier or this individual, okay, you can go back in now. You can take another hit. You can go back in and play. I do not have a good test that can definitively say when you can come back into play. I have to rely on the recommendations of both the Berlin Conference and the Zurich Conference and the stepwise process that all trainers are trained about. They follow what's called the Zachary-Lycett law, which I was involved with in the state of Washington, which states that you start them back into school. If they show symptoms, then you take them back. Then you take them out to let them do some exercise. If they don't show any symptoms, and the next day they can run around the track, the next day, and then finally they're clear and they're about ready to go. 80% of individuals, I was talking with the trainer at UNM here just before my talk, about 80% of the people clear in about 14 days after a single concussion. Then there's this miserable minority that seems to take months and sometimes years to recover. The third leg of the stool is when you tell somebody to retire. Got to stop. I'll never forget this. I was in Laguna Beach. I was at a conference for concussion, and I'm supposed to give a talk about concussion to these NFL players, linemen, and some quarterbacks. I tried as hard as I could, and I don't mean this derogatorily, but I tried to keep the science down and try to keep it matter of fact, that kind of stuff. And I'm sitting at lunch, and we have these sports writers, and I understand there's a sports writer here from the Journal, and I thanked him for coming. And they all wanted to talk to the quarterbacks and the football players. Troy Aikman, and Steve Young and Warren Moon, what's it like? How you doing? And then this guy turns to me, and he's from the Dallas newspaper, the Dallas Morning News, I think, sports writer. And at this particular time, the Raiders were still playing in Los Angeles. Okay, they were still the team in Los Angeles. And Troy Aikman said that he had had 9 concussions. So the sports writer turns to me and he said, "Would you recommend, Dr. Hovda, that Troy Aikman retire from the National Football League?" I said, my job is not to tell Troy Aikman when he should retire from the National Football League. My job is to tell Troy Aikman what his chances are, or what he could get if he continues to get blows to the head and plays in the National Football League.
What were his chances?
In my opinion, his chances of having long-term problems are going to be 40%. And he turns to me and he says, "You don't get it, do you, Doc?" And I said, what am I missing here? You don't know what it's like to play football in the National Football League. And I said, "No, I don't. I played football in high school, Sandy High School, or Comanche Junior, middle school, but I've never done the National Football League." He says, "I would give anything to play football." I said, "Would you give up the ability to recognize your wife? Would you give up the ability to recognize your grandchildren? Would you give up the ability to feed yourself?" And he said, "In a New York second, I would do that." And they all said that. And I don't know how to fight that, And when Paul Tagliabue was commissioner, he had me speak to all the owners and all the players, physicians, and we all talked about the risks and stuff about concussion. And the owner said, you know, we don't want to put this in the contracts because the disability clauses is going to cost a lot of money. And so then I went to the players union and I gave the same talk to the players union, and the players said, no, concussion doesn't happen in the National Football League. I said, what in the world? What is wrong with you guys? And then all of a sudden it says to me, I was told, you know something? These guys are paid on how they get paid the amount of money, how much they play. If a doctor takes them out for a week or 2 weeks of play, it could cost them $1 million. That costs a lot of money. Do you realize how much money's made on Monday Night Football? That's amazing. So I learned quite a bit. Yes, ma'am.
Once you have the energy crisis and cell
Yeah.
death, is there any way for the cells to recover?
Yes. In my professional, excuse me, in my professional opinion, when we've done this in animals and in humans, is that if we remove them from the field, whether there's it was a young woman that was playing lacrosse that she got a concussion, move her from the playing field, and went through the stepwise thing, and we let Mother Nature do what she wants to do. And then we slowly began to work this individual back into play. And then after about 3 or 4 weeks, she was fine. She went back into play. She was fine. So you know, think about this for a second. Teleologically, if we didn't recover from mild traumatic brain injury, teleologically speaking, several thousand or million years ago, we would have been somebody's breakfast. We wouldn't have recovered. We wouldn't have this ability. We've got this ability to recover. We as scientists or as physicians have just decided to kind of mess around with this a little bit and have actually done a few things that may be a little bit wrong. So when I started at UCLA, I turned to Don Becker and I said, you know, Don, I don't think we actually know what happens following traumatic brain injury. I think all these things that the pharmaceuticals want us to give to make money, they don't know either. That's why nothing's working. So we need to start from scratch. Now, concussion happens to both men and women. Women get concussions more often than men do. Well, why is that? Well, one is that you have a different anatomy. Okay, the French were right to some degree, okay? So you got a smaller head, a supple neck, so you get more violent movement of the brain. You also have this thing called hormones that occur differently in different cycles of the month. And it depends on when you have your concussion, that's important. So you actually respond differently. Now, you complain more often of symptoms following concussion than men do. Now, in my personal opinion, not professional, but personal opinion, is that I just think women are more honest. And I think the guys are just trying to cover it up. You know, women will actually say, listen, I'm not sleeping well and I've got these weird dreams, and that's the problem. There was a question back here first, a guy in a hat. Yeah.
Yes, so it sounds like concussions and, you know, brain injuries are kind of inherent with contact sports, I would say. So what do you think is going to be the future of such treatments, and is there anything to improve the situation at all?
Oh, I think there's lots of things to improve the situation. You have an example up here. The UNM football helmet, Kim, 1940, I don't know, the leather ones, 30 or something like that. And then we come up, and so the equipment is changing. So we're getting better equipment. The helmets, inside the helmets here are hard. There's a little give to it, but they're pretty hard. A helmet doesn't stop concussion, okay? It doesn't stop it. But players sometimes, if it's designed properly, it can reduce the amount of acceleration and deceleration. The rules have changed, okay? So now you can't spear with this before. I think that there are two kinds of sport. There are sports where head injury is part of the goal of the sport. Think of boxing, okay? The goal is to produce a concussion, okay? football, a concussion, or field hockey, or soccer, or something like that, concussion is a byproduct. And as we increase our awareness of when these things happen, the rules may change. And yes, we're going to reduce the amount of instances that it happens, but more importantly, it's going to happen regardless of what we do. It's going to happen. But we're getting smarter to the point to we're now able to recognize it and handle it. So we're not having a Long from the Pittsburgh Steelers committing suicide, or a Seau driving his car over the edge because he's gone crazy. I mean, or the battered wives, I think, are going to go down. I think we're going to improve that because we're recognizing there's a problem. It's now cool. I'll never forget this. There was a guy in Afghanistan, and I led a mission with 3 guys to Afghanistan. It was called Grey Team 3. And I was talking to a master sergeant, and an E6 was helping me. It's a corpsman. And he says, I had a concussion. I get to sit out for 2 weeks. I had the same thing Jay Cutler had when he was playing for the Chicago Bears. I've had a concussion. Isn't that cool? 20 years ago, he would have kept that silent. So, yes, ma'am.
So, in talking about, there's a lot of talking about how things happen and if there's any way of recovery, but is there experimental things out there that you're working with or that you actually trust? Or that, and that might be helpful in this situation? And also, how long would it take for an experimental, like, drug or method to come into practice?
Let me ask your middle question first. I was a graduate student here at UNM. It's 1979. And I was asked to give a lecture to the medical school class. It was a first-year medical school class. And I told the medical school class about this new thing that was coming out of Florida, out of the University of Florida, my colleague, that was called neurogenesis, that you were making new cells. And after the class, that I was called into the dean's office, and he said, "Don't you dare lie to our students." I said, "You got to be kidding. It was published in Science." He says, no, we're going to make these students pass the board. The board exams are going to say you don't make new cells. That's where the board's needs to be changed, all right? You do make new cells. Neurogenesis is one of those things I have hope for. I'm not positive yet. Scientists are naturally skeptical. I'm a skeptical person. I'm not sure every tweet that comes out of the White House is accurate. Okay? I'm just naturally that way, all right? I do think that there are things in the brain. I didn't get a chance to get into this because of time. I didn't want to bore you guys with boring things. But there are these things in the brain called neurotrophic factors or brain trophic factors. And so an example is if you take my brother's got 4 beautiful children. If his oldest son, when his oldest son was 2 or 1 year old or 2 years old, 3 years old, and we dropped him over in Paris, I picked him up, and about 2 months later, he's speaking French. You take me over to Paris, you pick me up in 3 months, I can't tell you how to find out where the toilet is. There's a lot of plasticity in the brain. We have just begun to tap it. And that has got great potential. And that comes, I can't give you BDNF. I can't give you trophic factors. But you can make it. And I can show you how to make it. And you make it, I'm afraid to admit it, my wife is right. I've been married 37 years. My wife's always right. I'm always wrong. Exercise the brain makes neurotrophic factors from exercise. So those are my big hopes. Yes?
Yes, I have a question about, you had spoken about noise being a factor to concussion.
Yeah.
Like in war,
Mm-hmm.
those large mortars
Yeah.
How is that how could that be a mild concussion?
Good question. When a cannon goes off, it's instantaneous. It's a loud, instantaneous blast. If you go to a movie concert or to a concert and they do a blast right away, it's the same. It can shock you. Okay. Usually, though, in the theater or in a movie concert, it's just loud, and your ears ring. You get headaches and stuff like that. I'm not, professionally now, I am not completely convinced that blast itself causes a brain injury. I'm not convinced yet. I've seen the data, and it's remarkable. In the autopsies that I've seen, the brain damage is in the posterior inferior part of the brain. I have no idea why it's there. I know that people are different after they've been exposed to blast. My dad served in Korea, and they used to call it blast syndrome or concussion syndrome. So I know people are different. I think what happens is that the blast causes what we now are starting to hear about, a subconcussive blow. A blow, subconcussive, which means it doesn't go through all of this stuff I'm telling you about, but it does make the brain a little more vulnerable, so that if it gets hit again, then it's a problem. So in Iraq and Afghanistan, I was surprised by the number of people that have concussions that are in a firefight. You can't pull a guy out or a girl out during the middle of a firefight. They got to stay. What I was worried about is the people that are traveling in an MRAP, and the MRAP hits an IED, and it gets rolled over. There's 7 people in the MRAP. Which one has the concussion? I'd like to know that. And that may be where the serum test comes in. So I may be able to do that. Yes.
Dr. Habda, I think we have about 2 more questions.
Okay, yeah, he's my boss, so I have to do what he says, sure.
Amateur boxing is doing away with headgear.
Mhm.
The open division fighters, I think it's age 16 and above.
Yeah.
The younger kids are still wearing them. Pro boxing, of course, has never used them. How effective, in your opinion, is headgear in preventing concussions in boxing?
None. Headgear is really good to stop contusions. So if I hit you with the side of the head with a helmet, no, I'm sorry. If I hit the side of your helmet with a hammer, you're protected because the helmet's there. But if I take, I always block on this guy's name. He was a NASCAR driver, number 3. Yeah. He died, not because he was hit in the head, but because the head was violently moved forward and back. He had a helmet on. So now they have a harness that they anchor it to the back. So we could do that in players. We could take the helmet, harness it to their shoulders so they can't move their head. Maybe that's why I didn't play football very well. So that would start it. There are studies now, today, I've been arguing for many years. I wanted people to look at the types of concussion and severity of concussions that you get in rugby, which doesn't have that kind of protection, and in football, that has protection. I'm hopeful that my colleagues in bioengineering, I'm not a bioengineer, has a way that will diffuse the force that would normally be that would normally permeate the skull into the brain, that would get diffused around the helmet and stop the concussion. I'm hopeful of that. I hate it. Bob Udall, the Udalls, when they were senators, there were a couple of companies that came out with helmets that they said stopped concussions, and I was asked, and I said no. I gave you the same answer. And I looked at the data and I said, no, doesn't happen. You can't make that statement. And so they stopped making it.
So Dr. Abda, as the last question, seeing as this is a Lobo living room and the majority of folks are alumni, and you as an alumni of this, of the University of New Mexico, 3 degrees, could you wrap up or finish maybe with your experience and how your, how you have impacted, which is very, very important, the military, the Department of Defense, and the impact that you've had in your work and your research has had on our United States military, and you know the reason that you have that medal on your chest, and why you've been recognized in that way, and how the military has been fundamentally changed when it comes to concussion, traumatic brain injury, removing soldiers from the field. If you could just explain that to us as an alum, we certainly are proud of you.
Yeah.
And would, and that's something that is quite lasting. So,
I'll pay you later, Patrick. I went to get my first grant, and I went to a board, and had all the deans of all the schools of UCLA, of UC, so UCLA, UC Davis, Berkeley, around the table. And the grant was being given by the Bank of California. And the rest of on the other side of the table were all the bank vice presidents. And at the head of the table was the president of the bank. And I had this grant, and couldn't show any slides, I could just tell them what the grant was about. And the guy from San Francisco looks at my CV and he says, Dr. Edward, we're very impressed with your application, but there's something weird about your CV. I said, what's that? He says, well, he says you started college at the University of New Mexico in 1971, and you graduated in 1979. What happened? And I said, well, I came home from school and after my 3rd year, and my dad says, son, you're wasting your time. Your grade point average is 1.8. I know you're playing golf all over the place. So I was playing golf. And you're having a great time. But this is a waste of time. I said, you're right, Dad, it's a waste of time. So I gave up school, and I tried to play professional golf. I played 1 round of golf with Nancy Lopez, and then I realized I can't make money doing this. So I went to the School of Political Science. And at the School of Political Science, I don't know where they are now, but they used to have a board, and I closed my eyes and I pointed, and that guy, I went to his office with his tie on, my best looking shirt, knocked on the door, and I said, all I need is somebody to sign a piece of paper, let me have 1 semester, let me make grades, and I'll stay. And so he signed, after he heard my story, he signed the piece of paper, and I stayed. And I got on double secret probation. And then graduate school was the same way, because my grade point average was 2.6 or something. And Dr. Delaney, who's sitting in the front row, he was grading one of our exams for statistics, and he grades them blindly. And he sees me in the hall, he says, is this yours? He says, yeah. He says, you passed, you can stay. And then they sent me to medical school here and graduate school, and of course I got my bachelor's degree, and then I went to UCLA for postdoctoral degree. For the military, I built the National Intrepid Center of Excellence with colleagues that treats mild traumatic brain injury and post-traumatic stress, and 10 satellites, of which on April the 4th, I'm dedicating the 8th one to the military at Camp Pendleton. I led a mission to Afghanistan to make sure that people were following the rules in terms of taking wounded warriors out of field of battle. I got a call when I was in Patrick's room from Admiral Mullen when Gabby Giffords was shot, to take care of Gabby Giffords in Tucson. I said, I'm not going to do that. I'm going to let the neurosurgeon take care of it, because I knew the neurosurgeon, and he does a good job. And I put together 2 blue ribbon panels to help individuals understand at the Joint Chiefs level what concussion was and why we stopped, why individuals should be protected, both in garrison and in theater. And then Admiral Mullen came by my office. He calls me and he says, "I want to have a secret meeting with you at UCLA." And I said, "Admiral Mullen, you're the chairman of the Joint Chiefs of Staff. You show up on campus with 4 black sedans, it's going to be really hard for me to keep the press quiet about this." And so he shows up and he says, "I got a mission for you." And he explains to me this mission, and we do this. And then he says, "Oh, and by the way, we want to celebrate your career." And in 2011, the United States Army has what they call the Tattoo Party, which is a parade party to celebrate the birthday of the Army. And every year, or every other year, they give the highest degree, highest award to a civilian from the Army for their service for their country, and that's what this medal is from 2011.